So I hope most of the participants are here again and we can continue with our second lecture. It is a lecture on topic fair image analysis, unveiling insights through reproducibility in image-based spatial transcriptomics. And the lecturer of this lecture is Tong, a senior software developer with a solid background in the field of image analysis, data science, and as I know, he's working in Bayraktar and has learned a lot about via image analysis, which currently will be introduced to you. So if you're ready, you can start.
Thank you very much, Yana, for the introduction. Thank you very much for having me here. And as Yana introduced, I'm a senior software developer working at the Wellcome Sanger Institute. And just a little bit of background about myself, because I've been told this in this audience, potentially a lot of students are here. So it might help you career path in the future if you want to do image analysis or single cell biology, etc. So my brief background is that I was trained as a biologist during my bachelor, purely biology. I didn't know a lot of code. And then I switched to bioinformatics during my master, and I didn't like sequencing because the major of my master was sequencing. So then I switched to bioimage analysis, which I love. And I've been working on this field for almost 10 years now. And that is the reason why I'm joining Sanger to developing image analysis pipelines for spatial transcriptomics. So enough about me. The topic of today is going to be fair image analysis. I will come back to what fair means in the downstream in the next slide. And the goal is to tell you what we've been doing in terms of image analysis at Sanger and why do we want to do fair image analysis. Okay. So before starting, the outline of this presentation is going to be split into 4 parts. We may or may not have a break in the middle. Just ask me if you want to have a break, potentially between the session 2 and 3. And first, I will just introduce to you what the image analysis that we are doing at Sanger Institute. And then I will just show you one specific examples of image analysis, a very typical image analysis that we are doing at Sanger Institute, which is called in situ sequencing image analysis, the ISS. And third part is going to be a little bit downstream of analysis, more like image data visualization, but for equitable data access across the world. Anybody in the world will be able to use our links to visualize this image data in addition to the sequencing data. And of course, in the end, take-home messages and future direction of what I want to do at Sanger Institute. So a very brief intro about what spatial transcriptomics is. Some of you may know, if you're in biology, you may know that spatial transcriptomics is a very hot topic these days, and it was even the Nature Method of the Year in 2020. And this is also demonstrated that if you look at the number of publications and number of data sets that with this spatial transcriptomics as tags, they are skyrocketing even nowadays. So the latest days here is just 2021, but in the recent days, this is even skyrocketing much higher than this one. So it's really, really hot topic. And there's no surprise that at Sanger Institute, we're interested in this as well. But that is very different, I would say. It's very different from classical sequencing analysis because that is just one dimension, AGCTs, and you align sequences, you find gene expression, profiles, etc. But with spatial, it's at least 2D and even 3D because you will be playing with 2D images or even 3D volumetric images. And for that, we need specific tools to do image analysis. And as you can see on the right, image analysis is, it can be represented as a pipeline, but if you just go into different steps, as I hopefully can see sorry, you cannot see my mouse, which is weird. Never mind. So image analysis pipelines are typically separated into a lot of steps. You do imaging, it's one of the steps, and then you do preprocessing, detection, segmentation, etc. You don't need to understand what all this means, but it is to tell you that image analysis can be relatively complex. It's not one-liner that you run and you get your result in the very end. And there's often a lot of feedback loops, because if you look at all the arrows, they are double-edged. It means that depending what you see in the next step, it might have an impact in the previous steps. So you may need to get back to your previous analysis. And the people who does that is usually a bioimage analyst. So this person should know more or less all the steps that we are talking about, because any of the steps can have an impact in the downstream analysis. So this is what we do at Sanger. So I'm currently the person sitting in the middle, and we do have small work groups, but that is really limited amount of the people. So in total, we have 3 people working on this image analysis at Sanger Institute. But knowing that Sanger is a relatively big institute, we have 6 programs, and 4 of them are also embracing image data analysis, including CellGen, where I come from, human genetics, cancer aging, parasite, and microbes. Those are all big programs. Each of them contains more than 100 people at least. And so you can imagine the quantity of work that is requested by the scientific programs, and there's a lot of things needs to be done. And if you also count in terms of data, we are generating almost 2 petabytes of data in the last three years, which doesn't seem to be a lot, but knowing that Sanger is relatively new in imaging, in the last 3 or 4 years, we were just setting up the imaging infrastructures, meaning that during the setup, we've already generated 2 petabytes. When everything runs in production, the speed will only be accelerated. And even during that construction stage, we have more than 50 projects ongoing in terms of image analysis. So the big question for us is that with a limited amount of people, or here maybe 4 or 5 people, 3 to 5 people, how can we support all the requests from all these programs? So our question, sorry, is there anyone? Never mind. So our solution is to use FAIR image analysis. So what FAIR means is findable, accessible, interoperable, and reusable. Basically means whatever we wrote, we want it to be accessible to anyone new to this field, and we'll be able to reuse it without much issue, without much hassle at least. And this is important in terms of engineering, but this is also important in terms of scientific discovery. Because if I may, I just going to read this definition of scientific discovery from the American Heritage Dictionary. So the definition is that the determination of existence, nature, or exact parameter of something by examination and interpretation of evidence obtained in many cases under controlled conditions that typically includes the use of hypothesis testing and rigorous statistical analysis, with the ultimate goal of producing results that are repeatable and reproducible. So by definition, all scientific discoveries has to be repeatable and reproducible. If they are not, they are actually even not scientific discoveries. Because if you can imagine, you take an apple and put it at height and you release it, it won't drop onto the ground, meaning that gravity is not scientific discovery because you cannot reproduce it, right? So for any other data analysis, it should be the same. Anybody who has the same controlled condition as stated in the definition, you should be able to reproduce whatever this scientific publication is talking about. So for that, we need to follow FAIR principle. This is about not only about actually data, but also about the tools that we are using. I'm talking about computational tools, so i.e. softwares. So for computational part, you already have a lot of tools. For example, people know GitHub, where you can put your open source code there, and anybody there can look at it and reproduce it, rerun it on their own. And that is a good way of making your software or your data findable and accessible. But I want to put a little bit emphasis on the reusability at the very end, because findable and accessible is just the first step of making everything reusable, and reproducible. So in order to make everything reusable, and you will need a lot of different tools and a lot of very controlled, very controlled way and very normalized ways so that you can do that. I won't be covered the interoperable very much in this talk simply because we're not doing that much in this pipeline in the downstream of my talk. So I'll just ignore it for now, but feel free to ask any questions about that if you want to. So as I mentioned, in order to make data analysis reusable, you will want to use workflows, and especially because image analysis is represented as a workflow. So just want to remind you the circle that I just showed to you, the definition of image analysis. And in order to do that, you have a lot of existing workflow managers already. So you have a Galaxy, you have a common workflow languages, Nextflow and SnakeMake. Those are the big players in the field. So I do know users of all of them. And somehow Nextflow is now dominating the domain. So especially in sequencing area, basically this is almost the only workflow managers people want to use, including Wellcome Sanger Institute. So we have the largest Nextflow communities around the world. We have more than 100 users using it only within the campus. And there's of course also Genelia, Francis Crick, ENBO, GSK, and ENBO EBI as well. So how do you choose amongst these big workflow managers, and which one is the best for you? So our goal is our rule is relatively simple. We want it to be language agnostic, meaning that whatever computational tools that you are using, it should be compatible with workflow manager, because we don't want to restrict people to write only Python code or only C++ code. It doesn't make sense. It has to be a language agnostic so that this whole infrastructure can be more flexible. Anybody writing anything can be reused in this framework. And of course, this has to be portable, meaning that this same piece of code should be running on your laptop, but also on Google Cloud or on the HPC system in your institute. And I'm talking about exactly the same piece of code. You shouldn't be changing a lot of things except the environment variables, etc. And last thing is the containerization. All the codes need to be, this workflow manager needs to support containerizations, meaning that you should be able to run Docker images and Singularity images just within this framework, and you don't need to think about further software installation problems, etc. So there's of course some other features. I'm not going to list of them. For example, cloud friendly and easy resume. These are the features that are really handy if you're a developer. But for us, the really three most important features that we need is the three that I just talked about. Now I will just dive into the analysis pipeline that I was talking about, which is called in situ sequencing image analysis pipeline. So this is a good example of showing what we've been doing. And at the very end of introducing each of the steps, I will tell you why we are talking about reusability of this pipeline. So again, a little bit of background of what spatial transcriptomic or in situ sequencing is. If you're a biologist, you will know that for decades, people has been putting biological samples onto the slide, as you can see in the middle. Then you just add different tags onto this slide, and you do imaging, and then you get the image data, as you can see on the middle right. So there you are looking at the DAPI in deep blue and some RNA spots in cyan. So, and then you do cyclic imaging. This is how in situ sequencing works. Basically, what it means is that you take one slide, you add your fluorescence tag, you do your imaging, and you strip off the tags, so the sample should become black again, and then you add another round of different RNA labelings, and you will be able to see different RNAs at the very same cells even. And then we do analysis to extract information from this cyclic imaging. More specifically, if you are biochemist, you'll be interested in this. but I will just go through it briefly. This is how the staining works. So on the top left, you see there's the slide. First, you do reverse transcription. You will have the mRNA expressed in large quantity, and then you put padlock probes, hybridization. So you will have that product fixed and then released from the mRNA. Then you do ligation and rolling circle amplification. The goal is to just increase this number of mRNA molecules massively so that you will be able to visualize it under microscope. Because if it's the only single copy, it's just too small, and the photon it won't be able to emit enough photons, and you will not be able to see it. So you have a lot of rolling circle products. So that is expression of these genes a lot of times. And then you can add fluorescence probes onto it. And as I mentioned before, you add one probe, you do imaging, you strip it, and you do another probe hybridization, imaging, and strip it. So just do the cyclic manipulations for several rounds. So each of the mRNA molecules will be encoded as a sequence of color. So I'm going to come back to this later when I talk about decoding of these colors. But here you just need to know that one gene will be encoded as a sequence of color. This sequence of color will be used as the ID of this specific gene. And then it was about the white lab, and then I will be mainly talking about the dry lab part because I'm, after all, software developer. I've been writing code for this. So here we are looking at the pipeline of this in situ sequencing image analysis pipeline. So you don't need to remember what these are at this moment. I just want to show you that if you pull these consensus of image analysis representations on the left, you will see a lot of matches between them. So we're just taking part of, so for this pipeline, we're just taking several steps and then make them a workflow, and then we run the things. So of course, there are many methods of pre-processing, many methods of peak calling, and many methods for single cell segmentation, but we are just using. But this whole workflow should be software agnostic as well. So for example, if we talk about cell segmentation, of course, they are state of the art, which is cell pulse. I'm going to introduce a little bit later. But if one day there's another segmentation method that is much better than cell pulse, this structure shouldn't change, which simply change what is the inside of these steps. And this is the way we make this whole pipeline modular. So enough about this. I will be just dive into each of the steps. The first one is called stitching. Sorry, the very middle one is called stitching. So for people who work with microscopes, potentially you all do during your bachelor. If you look into your microscope, you will see some cells. If you are using the correct magnification, you will see several cells within the field of view. But for whole slide imaging or for whole tissue imaging, typically you want to collate hundreds of thousands of the field of views together, because otherwise you won't see the big pictures as you can see on the top right. This step is called stitching. So we take images from both proprietary platform like PerkinElmer, but also bespoke microscope like the one in the middle, spinning disk confocal microscope. Both of them are just taking field of views, and then you need to stitch hundreds of thousands of field of views together to form one big image, as you can see on the right. And for that, we're using some open source softwares. In this specific case, we're using Ashlar. So the input of this step is to take field of views with 5 to 10% overlaps, because we need that overlap to calculate the translations between the adjacent field of views, and then we can stitch it back to one full image. And as you can see on the right, it works pretty well. And this image is like 50 gigs, and will take several hours, I think something like 1 hour to finish the stitching. And then starting from this stitched image, we'll do registration. remember that I told you with in situ sequencing, when we do cyclic imaging, meaning that the same sample will be imaged several times. And in between, we will take the sample out of the microscope. So when you put it back, there's a large chance that there will be a shift between the last position that you acquired. For that, we need to do a two-step correction, meaning that we want to align the same cell from two cycles. And this is called registration. For that, we use a tool called microaligner, which is developed at Sanger Institute. The first step, we just use the typical and classical computer vision tools called feature point registration, and that is based on SIFT. And if you know computer vision, you know what SIFT is a feature extraction step, and then you just align those feature points. And this is a rough translation, as you may imagine. There might be some distortions, and they are often not linear, meaning that a simple translation won't be able to fix won't be able to align cells at the subcellular level, as you can see in the middle. There are still some mismatches between different cycles. And then from there, we do the second step, which is called optical flow-based registration. So in this specific case, we really align pixel by pixel. And so you can see the improvement of the alignment quality at the very end on the right. So with that, we have the whole images. So the image processing is more or less done. And if you visualize this aligned images from different cycles, you will be able to retrieve really the same cell across different cycles. As you can see, they are, in terms of content of each single cell at different cycles, they look different, but simply because we are tagging different mRNAs in different cycles. So the goal is for each single dot in the images, you want to extract the peak profile, meaning that for one dot, it's a pity that you cannot see my mouse, but never mind. So you can imagine for one dot, it's just you want to see what is the sequence of color. In this case, the sequence is always going to be 7 elements. If it's red, blue, blue, something like that, what gene this corresponds to. And before doing that, actually, we also need to do cell segmentation because in situ sequencing is single cell analysis. First thing that you need to do is to find where the cells are. For that, we use a deep learning-based solution called CellPose, which is still, so this paper was published in 2021, but nowadays there's a recent review saying that this method is still state-of-the-art at this in the area, at least for bioimage analysis. So it's really pretty cool tool, and it not only applies to biological images. You can even try with. I played with it a little bit. I tried to play with rocks from just industrial applications. It does pretty good job. So you just can throw whatever images onto it. It will be able to segment blob-like objects like cells as segmented images on the left. And we just apply CellPose onto this whole section that I was talking about. In total, we have more than 92,000 nuclei detected. There's one trick is that CellPose is not meant to be very scalable at this large scale image analysis. So we had to do a tile-based analysis. Basically, we split these whole images into large tiles, and then so that it fits into the memory. And then we just stitch them back into one big images and do the downstream analysis. I mentioned about mRNAs detection. Sorry, I mentioned about decoding, but before decoding, you need to find where the spots are. This step is called peak calling. And in the very, very first version of this pipeline, we were using a software called TrackPy. As you can see on the left, without out of the box, TrackPy detects most of the spots, but also with a lot of false positives. So the red dots in the middle of the black area, they doesn't make sense. There's nothing there. So in order to use this tool correctly, you need to first do a pre-processing, which in our case, we just applied white height filtering. And then you will see you can see the detection of the right is much more accurate, and there's less false positive and less false negatives as well. So this is the classical way of doing peak calling. But nowadays there are some, again, deep learning tools. In this case, Spatial, which was released like 2 months ago. We've had a test run, and it seems to be very promising. And most importantly, with TrackPy, we had to manually define some threshold in order to make sure that peak calling is not giving too much false positive or too much false negatives. But with Spatial, as it's deep learning based, there's almost no parameters that you need to define within the program. So that is much promising, and that is helping making our pipeline much more reusable as well, because otherwise every time when you talk to biologists, so which threshold you need to use, otherwise your result might not be accurate as you expect. So finally, the decoding. As I mentioned, each gene will be represented as a sequence of color, as you can see on the top right. Gene X potentially, well, this is just a dummy example, but you can imagine gene X will be blue, a green, cyan, something like that. And if you see that sequence in each single of the dots that you detected, there's a very large chance that will be the gene that you're talking about, gene X, for example. If there's one mismatch, for example, if the cycle 3 is like red, potentially it will be gene Y, etc., but also with a probability. So what do we do? So how do we do that? In order to do that, we use Milana Gadarit from EBI. She wrote a post code, which is still in bioRxiv. Haven't been officially published yet, but we've been using that extensively already at Sanger Institute. In order to do this, we just extract the peak intensity across all cycles and run the code, and this postcode will tell you with this likelihood, this sequence of color belongs to gene X or Y, etc. And then that is how we assign colors for all the genes as you can see on the right. So each single dot is a mRNA, and the color represents different genes. So, and then now you can imagine we already have all the cells segmented, and we also have all the mRNA decoded. And then, so next thing, what we need to do is that we need to know which genes are in which cells. This is called assignment. You assign spot to cells. But you can imagine if you're having 92,000 nuclei and 2.4 million spots, and you run a for loop on it, it will be almost impossible. And computationally, it's just very, very slow. So luckily, like almost 30 years ago, some clever guys who invented this sort tile recursive trees. This will massively accelerate the assignments. So briefly, what it does is that it first, before running the assignment, it indexes all the cells or the molecules, whichever you want. You can do either way. So you first index them, and then you group them into bigger groups, and then smaller groups within this bigger group, and then even smaller groups within this bigger group. So you just do a hierarchical clustering of these cells or molecules, and then when you query cells or molecules, you'll be searched only first at the very top level. Is this in this large region? If it is, let's continue dive in depth into this same region. If not, let's skip to another big region. So this will massively accelerate the assignment from days of running. So why I put forever here on the left is simply because I've tried to run the for loop. I think it ran for 3 days and didn't finish. So I just abandoned it, I killed it, and then switched to the other methods on the right. I think it took less than 1 hour to finish the assignment. This is purely computational, but it's a clever way of doing exactly the same thing. So this pipeline, of course, has been used within Sanger massively and had been involved in more than 10 projects. Some of them are still in revision or in manuscript preparation, but some of them are already out, as you can see the ones that are listed here. And there's human embryo project. In the middle is the human bone project. We're just looking at the gene expression in different tissues. And on the right is the human reproductive system as well. And so I'm not going to dive into any of this. The reason I'm putting here is that, as you can see, the same program can be run on different tissues and it still works pretty well. And you don't, to be honest, I didn't even change much parameters. I was just reusing the same parameters and it works for different tissues. So it's pretty awesome. So last thing that I want to talk about in this part is that what is a fair thing in this pipeline? So fair for this is a reusability. I'm putting this representation of my pipeline again here because I want to show you actually almost each of the components, each of the rectangles here are reusable, can be reusable in other projects. For example, on the very left, the stitching, for almost any imaging project, you will want to stitch your data back into a biological meaningful format because otherwise, well, there's technically there's no differences between analyzing each single field of view or analyzing the whole stitched image. But the biologists will always want to put everything back into its original position, so you will be able to see its spatial distribution, which genes are close to which ones. So stitching is almost always the case, and that is needed. And it will be reused in a lot of projects, not just in this pipeline. It can be for potentially organoid imaging, as I'm going to mention on the top right. And the registration is for aligning different images. So if they are from the same microscope, that will be great, but it doesn't limit to that. It can be multimodal, meaning that same tissue, if it's imaged with different technologies, theoretically they're just a different representation of the same biological sample. Theoretically, you should be able to register them together back into one data set, right? So registration can be reused in other multimodal projects as well, but we don't really have it yet. So it's just on our blueprint. But a lot of people are working on it. I'm aware of that. So this part will be reusable, and we're just waiting for project. For segmentation, as you can imagine, for any single cell or in any cell biologist will always be interested in single cell, even subcellular resolution. So cell segmentation will always be reused in whichever project as long as single cell. So, and we are currently even working on the organoid data, which coincidentally to be the natural method of the year again for last year. So we've been somehow keep track of the trend. And for the decoding part, I've just mentioned that is a decoding for in situ sequencing, but as this whole thing is open source, it's flexible, we were able to adapt it to Murfish-like decoding as well, which is just another spatial technology and use different ways of coding your mRNA genes. As you can see, these 4 steps are already reusable, potentially in other projects. And the very last thing is on the right is the visualization, which I didn't cover much in this step because that is the major content that I'm going to present in the next step, which is called Web Atlas. And this will, before going into that part, I would just want to acknowledge the people who were involved in the first project, and including my PI, former director, and Sarah Techman, who provided the data, and the Georgetown lab who did the post-code analysis, post-code software development, and our IT team for it, of course, always. And thank you, Maz, for providing some insightful helps on this. So now, I'm not sure whether we should take a break, but if not, I'm going to continue.
Well, okay, I think we can answer the questions which are already in Slido. I can share my screen with these questions.
Yeah.
Yes.
Should I stop sharing?
I think
Okay.
it's okay. Yes. Yes. So the first question is how can image-based spatial transcriptomics be used to analyze gene expression patterns?
Uh, what do you mean? So the goal of using spatial transcriptomics is actually to look at the gene expression because we are looking at the expression of mRNA molecules. You will be able to count how many mRNAs expressed in which cell, and also keeping the spatial information as well.
Okay, the second question which we had is what is the computational scale of this image analysis pipelines? And can they be done on a personal laptop or they are limited to large clusters only?
Yeah, good question. Actually, the image analysis part is actually even simple, I would say. Most of my work was spent on making the scalable. So I would say it's scalable, but it's not infinitely scalable because currently we are talking about data set as large as 200, 300 gigabytes. Well, I'm not talking about we need all that memories. In terms of memory, I think we still need at least around 50 gigs. So you may need a workstation or a HPC. A, it depends a little bit on the size of your data set. If your data set is large, you will need workstation. But if it's small, it's not human uh, it's not human tissue, you might be able to run it on a laptop. It really depends on the size of the data. And it really depends on the imaging method as well, because now we're just talking about 2D image data analysis. In the future, we might have a lightsheet 3D image data, and that is at terabyte level. I'm not sure whether anyone can do that on a laptop, but if with enough optimization, theoretically it is possible. Just people didn't have time to do the optimization yet because, as I said, spatial is quite hot. Biologists want to sprint to get the publication out, and then people will think about optimization, how to write on a laptop, etc. It will come.
Okay, then I can mark it as answered, I think. The next question is how does the resolution and scale of imaging techniques used in spatial transcriptomics impact the level of accuracy? And is there any limitations?
So here I'm just presenting one single technique, which is called in situ sequencing. But as people working in this area, you will see there are a lot of different technologies doing almost the same thing. And of course, one of the big discrepancies between them is the resolution. So some are single cell, like really image-based spatial technology are often single cell, like in situ sequencing, and also Xenium, and also Murphish. Those are the single cell technology. But there's also Visium and StereoSeq. Those are array-based sequencing spatial transcriptomic techniques. Those are the resolution will be roughly 15 microns in terms of diameter of one Visium spot. A StereoSeq is subcellular, so it will be even with higher resolution than single cell. And then the limitation is like number of plex as well. When I think number of plex, meaning the number of genes that you can look at in each single run. For example, in situ sequencing, we typically talk about for one single multi-cycle run that I presented before, you should be able to look at the expression of 200 to 300 genes. That is practically the maximum that I've seen. And same for Xenium, but there are some other techniques. I think Xenium, which is the commercialized solution, they claim that in the next release, we will be able to see 5,000 genes in one single run. But we haven't seen it yet, but typically that is one of the limitations as well. So if you're looking at a lot of genes, the only option for you might be just Xenium if you're just interested in hundreds of genes. In situ sequencing, Murphy-ish, those should be good enough. Hope that answers your question.
Okay, and the last one. For now, is it fair to say that deep learning tools are more accurate than traditional computer vision algorithms when applied to spatial transcriptomics data?
Really depends on the data we are talking about. So if we specifically talking about spatial transcriptomic data, meaning that is almost equal to large data set, in that case, I would say yes, deep learning is outperforming and is more accurate because I mean, just practically, I haven't seen any project hired one person annotating all the single cell data. So can we see really, I mean, sorry. So the answer is yes, because we're comparing deep learning with traditional computer vision. The traditional computer vision cannot handle all the complexities with one single images as the spatial transcriptomics. There are a lot of variabilities between different regions, and applying, for example, thresholding like O2 threshold, it won't be generalized. It won't be generalizable enough to segment all cells correctly. In the current state, yes.
Okay, one more question appeared. Does Xenium provide a proprietary software that performs the needed pipeline steps? And if yes, is it more accurate than the solution using open source tools?
So first part of the question, yes, Xenium does provide proprietary software, but that is even integrated into their machine. So you don't even have access to it. Everything runs as a black box. And the second part is that, is it more accurate? We cannot compare simply because 10x is not even giving you the raw data for the different cycles. So for example, in the situ sequencing, we still have the access to the raw data from different cycles. So we can do the decoding our own. But Xenium is not providing any of those data. They are just giving you the final analysis result. So you will not be able to compare to whatever they done before because you don't even have the raw data.
Okay, thank you for the answers. Then we can return to the second part of the lecture, and maybe we have more questions in the end too.
Yep. Yep. So let me share my screen again. Here it is. Where's my mouse? Which is weird. Okay, I don't see my mouse. Never mind. So the next part I'm going to talk about is not really image analysis. It's more about image data visualization and equitable data access. So by using Web Atlas, which is just a screenshot of Web Atlas, as you can see on the left, and it is a web page. So in few slides, you will be able to have access to it on your own as well. So as you might aware, the human human genomics project was initiated at Sanger Institute. So we have a long story of working with single cell data, especially single cell RNA-seq data. And now with a different method of spatial transcriptomics, those are the tools that are complementary to each other. So because spatial transcriptomic data has the spatial information in X and Y and where they are from the tissue. So in order to put all this data together, we want to have a holistic and global view of what is happening within human bodies. And in this specific case, the initiative, the motivation that we developed Web Atlas is because we have a project which is whole embryo project, as shown on the left, where we are generating data from multiple modalities, including single cell RNA-seq, Visium and Xenium, and in situ sequencing. Oh, sorry, not in situ sequencing in that case. It's just Visium, Xenium, and single cell. we want to visualize all this data in one same portal and be able to combine the strengths of different technologies to see whether there's more biological hidden interesting discoveries that is there. So as you might see, the advantage of single-cell RNA-seq is that it has a whole transcriptomics view. So you can, for each single cell, you can see the gene expression of the whole transcriptomics, but it doesn't have the spatial information. So for that, we need image-based or sequencing-based spatial transcriptomics. That, as I mentioned before, is going to be either Xenium or Visium. And so I've probably answered the question where I'm going to present here is like image-based has single-cell resolution, sequencing-based has a whole transcriptome discovery, so you're not limited to just 300 or 5,000 genes. You can really look at the whole panels. But the sequencing-based ones don't have the single-cell resolution. So the way we are thinking about combining it is to put them, to link the data and then put them into one single portal. How we did that is that we wrote an Nextflow pipeline again, taking different data set from different technologies, do the data conversion needed, and do the integration between those technologies needed, and then output a standardized ZAR format, which is optimized for visualization of this type of data. I don't think I will dive into how the conversion works, how the integration works, because that is going to be too technical. I don't think this not really needed in this talk. So feel free to ask questions if you are interested, and I can also send you the paper or documentation if you need this kind of tools. But I will just give you a global overview of what this does. So it is a Nextflow pipeline, a one-liner, as I mentioned, do data integration, conversion, and then give it, and then send you a link. Then you can visualize your both spatial transcriptome data along with the sequencing data. We actually, no, let me just take a step back. This paper was recently accepted in Nature Method, but as you can see, Nextflow pipeline is a computational tool. A lot of biologists don't really know how to use it. So the editor asked us, can you just do something even simpler? So we just did that. Martin Pritch from Celgene IT, actually head of Celgene IT, he wrote a web portal where there's web atlas running behind the scene. You just need to drag and drop your single cell and spatial data onto the portal, and then click run, click upload test, test the data set, and you wait for 10 minutes because there will be web atlas programs running behind. You wait a little bit, roughly 10 minutes, and then you will be receiving a link having the access your data. And so we've had this working. Unfortunately, I forgot to put the link here, but you theoretically should be able to play with it. Let me know if you are interested. But you can have access to the data here. I may just pause here for maybe 20 seconds. If you just take out your phone, scan this QR code on the left, you'll be able to see the data on the right. So this is the way we are democratizing our data from Sanger to the rest of the world. So this is the data, as I mentioned, it's a human embryo project. Oh, this data is already published, so there's no ethical issues. So feel free to use it and even spread it on X or Twitter or whichever you want. And I'm going to switch back one slide. So that is what I was talking about is just about visualization, but visualization doesn't really give any scientific discoveries, right? So it's a tool to facilitate data visualization, and then some people with expertise will be able to use this tool to dive into the data and to actually make real scientific discoveries. For example, in this case, Sid Lawrence from Sanger Institute as well, who was clinical PhD student. I think he just finished his PhD. So he used a web atlas And there's annotation tool within WebAtlas. He annotated all the digits within the handling and all the fibula and tibia regions. And then we will have these cells annotated with a manually assigned class when we do the plots on the right, because I remember that we have not only the spatial data, but also the gene expression data behind. So you can do the cell type compositions within these regions. And when you see the composition, you will be able to see that if you look at the proximal region, which is tibia and fibula, there's more mature cells, as you can see on the right. So there with the red arrows, and there is a gradient starting from tibia to fibula and no digits. So there's a developmental stage shift from the proximal to the distal region, meaning that there's more and more progenitor young cells on the digital side, and there's more and more old cells or mature cells on the proximal side. And this is something, even though we think, oh, this doesn't make sense because the tibia, and fibula are developed before the digits because there has to be a progression. But this hasn't been shown before. This is really this is one of the first examples that we really demonstrated scientifically. There is a progression and there is a gradient. Only by using this kind of tools and where we can combine single cell and spatial data. So I don't know if people manage to play with this link, but just want to remind you that everything is you can move everything within this portal. So the different panels are widgets. You can drag and drop. You can zoom in and out, and all you are looking at are the raw data. They are not a snapshot of your data and like using matplotlib, etc. And actually, if you go to the web link, you will see it is first prefixed with WebAtlas, and then the second part is HTTP to another config file. That is just a config file for visualization, but you will also be able to find the link to really the raw data. So if you want to reanalyze the data, you want to have access to the pixel data, so the in situ sequencing or single cell sequencing, whichever you want, you can find the links within that configuration file and reproduce whatever you want. So this is a way that we want to ensure reusability and reproducibility of our data analysis. So originally, this WebAtlas portal was not just built for just visualizing these three different modalities. Ultimately, the goal is to build even a bigger components on top of WebAtlas so that we can scheme the whole embryo data set, meaning that so on the left, we have the whole embryo. In this project, we are slicing it into 50 sections. And imagine you have one WebAtlas link to each of the section, and then you can browse across them. So you will really have a holistic view of the whole embryo in a 3D, well, in a studio 3D mode. And you'll be able to see the expression of each single gene or each single wisdom spot across the whole embryo. So this is a current effort that is developed by Dave Horsfall and Daniela from Newcastle University in Maz Hanifa's lab. And with that, I want to thank all the people who were involved in this part. So including again, Omer, of course, and Celgene IT, Maz Hanifa and Dave and Daniela particularly, because they are actually Dave and Daniela also the co-first authors of this piece of work. And of course, data provided by Sarah Teichmann lab, and also very insightful comments from open microscopy environments. So the last part will be take-home messages and the future direction of what we want to do at Sanger Institute, and personally what I want to do, because I will be starting a unit. Well, we're preparing for a bioimaging analysis unit at Sanger Institute because it is not yet officially there because Sanger is relatively new, but we are building toward it. So I will just tell you what my vision will be. So first, the conclusion, what I've been talking about. So we use Nextflow as workflow manager. we run image data analysis, specifically C2 sequencing, but we do run many other projects surrounding this. This is just a good representation of all the tasks that we do, but on top of each, there will be another project attached to it. So the way we run it, that we make the pipeline modular, and so that it will be a variable type of analysis possible with spatial data or single cell data. And we use Web Atlas for data visualization. It is currently just for whole slide imaging, but we also have other ways of visualizing data using Omero, which I will be briefly covered in the future. In the next part. So the future direction, I will come back to the reusability, as I mentioned. Since there's only a limited amount of components, well, it's a lot of ways of doing, for example, cell segmentation, but the number of type of task is limited. There are just several of them. Ideally, we will want to have all those steps and make them universally reusable, embedded into a software, embedded into a workflow manager such as Nextflow. Because other than that, I haven't seen something that can be as portable as Nextflow in other components. For example, Fiji is great too. The reason I'm not mentioning it is because it does have these limitations in terms of whole slide image analysis or high throughput image analysis. It is not made for that. It is great to play with small images. If you have just 2000 by 2000 pixels, which fits into your laptop memory, that is great. But for HDSG, for basically the department I'm working in is the whole high throughput spatial genomics. So the high throughput part cannot be handled by FPG. So that's the reason I'm not mentioning it. So we want everything to be universally applicable. Especially, it should be runnable in different labs, which has HPCs or slot clusters, etc. And we want these components can be used in different technologies. For example, the decoding should not be focused on just in situ sequencing for Merfish. It should be working as well, and of course in a scalable way. And ultimately, we will want everything to be runnable on a laptop. But it is already the case if your data is small enough. But as I mentioned, if your data is large, there's unfortunately still a little bit optimization that we need to do. Actually, when we talk about pipeline, usually there's one assumption that I keep omitting since the very beginning, but which is really important, which is the data format. You write a pipeline, the most important part is what is I/O of each step. For example, stitching here, in our case, we're still using STIFs, which is old file format. It's been there for almost 50 years. People has been working with it. Until now, they are very happy, but we are at the kind of next generation image format already because the image data is getting larger and larger. And with the rise the community of machine learning, there will be people will want parallelized image data, read and write. Those kind of things are not really TIFF friendly. So we need to switch from TIFF to some other things that are more next generation. So in this case, it's going to be Omizar. So as Sanger, we're trying to embrace Omizar as much as possible so that all the components that we write, as long as you're using Omizar, you should be able to write without worrying anything, right? And it has already been demonstrated that Omizar does have some benefits in terms of computation, because if you look at the example on the bottom left, when we run a deconvolution, which is the step before the stitching, so you need to correct your field of views a little bit using deconvolution. If you use OmiTIFF, that is going to take 2 days. And if you use Omizar, which allows parallelized read and write and computational processing, it will take only 45 minutes for the same data set. So it's a dramatic change in terms of performance. So ZAR has recently been published for both image data analysis, for both image data visualization and analysis. And as you can see on the paper on the left, there are actually more than 40 more than 40 authors of this paper, so a lot of people are embracing this new format. And we have Omero Viewer. It is now also supporting OmniZar that is running at Sanger Institute. So I mentioned several times next flows, and simply because in my limit field of view, so I do see it as the future of image analysis, at least for the large scale hash ones, because we're not the only one embracing this tool. There are other campuses, as I listed on the left. People are interested in converting or wrapping their data analysis tools into Nextflow. There's a joint effort on GitHub. If you want, just search bioimage tools. That is organization. People are just posting OmniZar related stuffs there. And we've also wrote OmniZar image analysis Nextflow dummy example. If people are interested, can just have a look. And this is for telling people what are the current state of art ways of processing OmniZar. But we need actually the current situation is that we need more people using it, and then we will have more focuses, and people will not complain about some issues of the format. There are still some minor issues which I won't be covering here today. One last thing, that if you are interested in any of the things that I was talking about, we're also hiring at Sanger Institute. This is going to be an image data scientist position. It's going to be starting with 2 years, I would say. But the job advert is not out now. Feel free to reach out if you're interested. And with that, thank you for your attention.
Okay, thank you. As I see, we don't have any new questions right now, but maybe we'll have them in a couple of moments. Okay, then to sum up some moments, the question from me.
Yeah.
Does now the integration of that multimodal imaging data, for example, combining spatial transcriptomics with some mass spectrometry or etc., help really help to understand some cellular interactions, and if this benefit is really significant?
So the short answer, well, we haven't done that yet. It is going to be very reckless to say that it will help, but I don't see any reason why it won't help, because with the Human Cell Atlas initiatives, which is a global effort, people simply want to know more and more about each single cell. We want to see from the same. For example, now we're interested in the sequencing data from single cell, and with the development of technologies, we're now able to see the spatial localization of each single cell along with this gene expression. And people are just crazy for it. It is the reason why this spatial transcriptomic is so hot. You simply added one dimension, which is the spatial information, and people are crazy. And imagine if you can add more modalities. And if you are aware, if you know that there are also Raman microscopy and also the ones you mentioned, mass spectrals, if you just add more and more dimensions, talking about same single cell, I'm pretty sure there will be new discoveries. But as I said, I haven't seen much combinations of that and showing that is a dramatic breakthrough of the scientific discovery. So I think we should just wait for it.
Okay, thank you. And are there any emerging trends, maybe in image-based spatial transcriptomics technology, that you find particularly promising?
Do you mean any technologies, any new technologies will be replacing potentially, right?
Maybe yes.
I think state of the art is now still Xenium. As far as I know, there will be Visium HD. I think the trend is just going to get higher and higher resolution and more gene expression for each single cell. That is general trend. And then the other trend is multimodal, because there will be a limitation. And after all, if you think about Xenium can, if it works, it's going to be 5,000 genes for each single cell. But when people do single cell analysis, they don't really use all those gene expressions, because typically what they need to do first is to do PCA or UMAP to find which are the most represented gene expression profiles, and then get rid of the rest of it. So whole transcriptome is useful, but you don't really, well, it helps you to narrow your focus, and then you can start your real analysis from there. So higher and higher plex, higher and higher gene expression is not really the mainstream. Well, it is required now. People think it will be useful, but I don't think it's going to be the major contributions of the scientific discovery in the future. I think the real benefits, the real trend should be multimodal, combining different technologies at the spatial scale. That will give a lot of more informative results.
Okay, thank you. We have one more question in Slido. Are there any alternatives to Web Atlas to compare its performance to?
Thanks. So, I didn't mention there are indeed some other frameworks that can do spatial data visualization, which the most known one is called Tissue Maps 3. And so I'm not sure what compares performance. Well, I'm not sure what does it mean, because if it's computational performance or the ability to different ways of visualizing your data. So if it's about the computational part, I don't think it's not comparable, because Web Atlas is after all serverless, meaning that it is just a stable, it is just a static, sorry, it's just static HTML file. All the computation will be happening on your browser, and all the web page does is streaming your data, fetch the data from S3 buckets to your local browser, and then it is your local browser who does the computation. So you drag and drop, zoom in and out, though it's all using your local resource. But for tissue maps, you do need to run a server. So there will be a computational backend, and all the computation will happen there. But it also does a little bit more than what WebArts does, because it can do some kind of clusterings, on-the-fly clusterings with it, and a little bit more dynamics with tissue maps. But yeah, hope that answers the question. So if that's what this question means.
Okay, I think yes. Then we have all questions answered for now. We have a little bit more time booked for this lecture, but if all questions answered, then I was very glad to hear your lecture, to listen to your lecture, and I hope that all of the participants also gained lots of new knowledge from it. And thank you.
Thank you very much.